Okay, I'm going to start here at transcription and translation. In some classes, I have already done this, so if that's the case, go ahead and fast forward through this, like first hour, for instance. So transcription and translation are the two main parts of protein synthesis. So it's going from gene to protein. So transcription is the synthesis of mRNA using DNA as the template. All right, so M stands for the message. Then translation is taking that message in mRNA. It's the process that occurs when the mRNA reaches the ribosome and protein synthesis occurs. And so when we look at this, in prokaryotic cells versus eukaryotic cells. So different things happen in prokaryotic versus eukaryotic cells. So, but they both have transcription and translation. So transcription is, here's the DNA. Notice the prokaryotic cell does not have a nucleus, so the DNA is here out in the cytoplasm. The DNA is used to make your mRNA, and remember, it's only the gene that's the DNA. So it may not be the whole strand of DNA. It's just, let's say, from here to here is the gene, and so therefore you make the mRNA from that gene in your DNA. That mRNA then carries those instructions to make your protein to your ribosome, which is the site of protein synthesis, and your mRNA is quote-unquote read, and these little dots here, each one stands for a different amino acid, which is what a polypeptide is, so it's a chain of amino acids. Versus in eukaryotic cells, so that was transcription and translation. In eukaryotic cells, the DNA is enclosed in the nucleus, and if you remember, DNA does not leave the nucleus, and so what happens is transcription, where we make a mRNA molecule from the DNA. Now notice here there's a second step that's in this process that wasn't in eukaryotic cells, and this is RNA processing. So notice that they call this the pre-mRNA, and here's the final mRNA. So a couple things happen here. Notice it's shorter, so we'll talk more about this later. It's shorter, so we cut out some of the mRNA. And what you can't see here is that, let me get a different color here, they put on a cap and a tail. We'll talk more about that later, but this cap and the tail, what they do is protect the mRNA strand when it leaves the nucleus from enzymes that might break it down in the cytoplasm. So it's a protection measurement, and that final mRNA can leave through the nuclear pore and go to the ribosome where it can be quote-unquote read. So here's the cap and the tail, quote-unquote read, and hook the appropriate amino acids to make the protein. And that's translation. You're translating that message into making a polypeptide chain. So that's the overview. So we're going to start looking at, specifically transcription here in just a minute. But before we do that, we have to understand a couple of things. We turn the page. This gives you an overview of what's happening. And so if you are looking at one gene, a gene is just a segment of a larger DNA molecule. So here's one gene, two genes, three genes. So we're looking at just gene one. So we're looking at just one side of the DNA. Remember, DNA is double-stranded. We use just one side of the DNA. And here's your bases sticking out. So this line here represents the phosphate sugar backbone. Notice here the DNA, we're going from 3 to 5 prime. So it's going from 3 to 5 prime, and that's because we're going to make an mRNA strand from this. So we're using this as a template, and we're going to make an mRNA strand. That's transcription. That's the first step. So if you notice here the mRNA, notice the bases in corresponding with the DNA. Do you notice that they're complementary bases? And so here's the A, and remember mRNA, they don't have T's. Usually T's go with an A, but mRNA instead of T's have U's. So this U matches up with the A, then we put a G that goes with the C, and so on. And so this is the coded message of how to make your protein from the DNA strand. It's based upon the complementary bases. And so if you look at, and if you were given an mRNA strand, could you tell what the DNA template strand was? Absolutely, because it would have the complementary bases. Now, why we're looking at the DNA strand from 3 to 5 is because just like when we make DNA, when we do DNA replication, mRNA is made, the new strand, in the 5 to 3' direction. So therefore, in order for it to make mRNA being made from left to right here, making this strand here in the 5 to 3' direction, this has to be the 3' of the DNA strand, just like DNA replication. Then, that's transcription. Then the mRNA goes to the ribosome to make your protein, which, so we go from mRNA to protein, that's called translation. So how do we know what proteins to make or what polypeptide to make? Well, remember, it's a chain of amino acids. So we quote unquote read the mRNA in groups of 3. So every group of 3 is called a codon. And so UGG, that codon codes for a particular amino acid. And so therefore that tells it the ribosome to bring the amino acid tryptophan. TRP stands for tryptophan. Then the second codon, UUU, that stands for phenylalanine, and so on and so forth. And that's how the ribosome knows what amino acids to hook together, is from the code from the mRNA, which originated at the DNA. So that's transcription and translation. So to understand this properly, we need to understand RNA. So RNA is used because it is a way to protect the DNA from possible damage. The DNA is nice and protected in the nucleus. It never leaves. The RNA does. Many copies of RNA can be made from one gene, thus allowing many copies of a protein to be made simultaneously. So what happens is when you're looking at one gene, it's not just made one mRNA at a time. You can make many copies of mRNA from that one gene, and all of those copies go to a ribosome and make lots of protein at the same time. Another thing that increases the speed which we can make proteins. Each RNA transcript can be translated repeatedly via a polyribosome. Poly means many, ribosome, so we're using many ribosomes. So what is a polyribosome? So here you can see, here meaning we're going to look at a picture in just a minute, you can see an mRNA transcript being translated into many copies of a protein by multiple ribosomes in a eukaryote. This is a way in which the cell can efficiently make numerous copies of proteins. This process is essentially the same between prokaryotes and eukaryotes. And so if we look at the next picture, let's flip this over here. Notice here, this is your mRNA strand. So here's your start of your mRNA. This is your mRNA strand right here. Start and end of your mRNA. Notice here that multiple ribosomes are working on this. So what happens, we're going to learn in just a minute, a little bit here, that ribosomes are made out of subunits. They come together, so this is a full, complete ribosome, and it starts reading the mRNA strand and putting together and building your polypeptides. So these guys right here are your growing polypeptide strands. So what happens here is that this ribosome just started and therefore has a shorter polypeptide strand. But this ribosome right here started here and has gone longer, so it's been adding more amino acids, so it's a little bit longer. This one started before that, so now it's much longer. And this one is just about to the end, and it has completed its polypeptide strand. And notice here, once the ribosome gets to the end, it dismantles and comes apart again until it's used again. And so that's what that means, that you can get many copies. You have one mRNA, and multiple ribosomes can be working on it at the same time. And then imagine if we had many copies of that mRNA strand. So you have lots of different copies in the cell, and you have lots of different ribosomes working on each one of those copies of the mRNA. You can get lots of copies of your protein that you need pretty quickly as a result. And this is an actual real electron micrograph picture showing one strand of mRNA. So the mRNA would be going through here with multiple ribosomes making the peptides from that. All right, so both prokaryotic cells and eukaryotic cells do that. But one main difference between them, between prokaryotic and eukaryotic cells, There's the difference between transcription and translation is that the two processes can occur simultaneously. In prokaryotes, because they lack what do they lack? A nucleus. So therefore, everything's happening at kind of the same time here in prokaryotic cells. So they can make your mRNA and then go right to making the protein and happen at the same time, versus in eukaryotic cells, the two processes must occur at different times. You have to make the mRNA first, because that happens in the nucleus. So transcription occurs in the nucleus, so the making of the mRNA, and translation occurs in the cytoplasm, so therefore they have to occur at two different times. The mRNA has to be made first, and then the protein second, because it has to leave the nucleus. The mRNA has to leave the nucleus and go to the ribosome, and so there's a order here that needs to take place, versus in a eukaryote or prokaryotic cell, when the mRNA is made, then it can immediately start being translated into a protein. So how is it that we use the genetic code in our cells to make proteins? So let's talk about the genetic code. So scientists began wondering how the genetic information contained within DNA instructed the formation of proteins. How could only 4 different base pairs in DNA, A, T, C, and G, how is only 4 base pairs code for 20 different amino acids? Because there's 20 different amino acids that are used to make your polypeptides. And so they found out that 61 out of the 64 codons code for amino acids. So we're going to write here code for amino acids. 3 of those codons code for stop codons, and which signals the end to translation, which means stop building your protein, and AUG is your start codon. And so let's look at this chart here, and let me explain how to use it. So here are your 64 codons in here. And so how do we know that a specific codon codes for a particular amino acid? So let's say here's your DNA. Here's your DNA molecule. And let's say the first 3 bases are AUG. Then let's say TGA. And then we have, let's say, GTC. And then we'll go ATT. So let's say that's your DNA. So in translation, which is making an mRNA from that DNA. So this occurs in the nucleus. So to make the mRNA from this, I'm going to have you go ahead and, so this is one side of the DNA from the 3 to the 5' end. So pause this and go ahead and write your mRNA strand that would be made from that DNA. So the mRNA is going to be made in the 5 to 3' direction. So pause it and do that. Okay, so you should have UAC. Oh, you know what? I got ahead of myself. Go back and let's redo this. This is not supposed to be AUC. This was supposed to be TAC. All right. So that was my fault. That was supposed to give you the first mRNA, which would be AUG. So that was my fault. Did you guys catch that? That DNA doesn't have U? All right. That should have given you your first clue that I made a mistake. My lights just went out. Okay, so AUG, then we have ACU, then CAG, and then TAA. All right, with that. Oh, sorry, not TUAA. because it's mRNA. They don't have T's, and so they have U's instead of T's. So that is your mRNA strand. So then what we do is then to do translation, which is using the mRNA strand at the ribosome to make your polypeptide. I'll just abbreviate for polypeptide. So how do we figure out what amino acid is going to be brought first? So we read the mRNA in groups of 3 called codons. So here's your first group of 3, second group of 3, third group of 3, fourth group of 3. Okay? So then what happens is we look at this first codon, AUG. So we come over here. And notice that this says the first mRNA base. So here are your 4 bases. So your first base is somewhere in this row. Notice here, second mRNA base. So our second here is U. So here are your 4 choices. Here's U. You come down here, and where they meet, we know that our codon is in this box here, the A box and the U box. And then we go here, notice that the third base in that particular row, there's a G, A, C, and U. So our third base is G. And so we go here to the G. Here's our AUG. And next to that, next to the codon, it tells you what amino acid stands for. So that's the first amino acid is methionine, or it says start. So this oftentimes your first amino acid in a polypeptide is going to be methionine because that's your start codon. That's how on your DNA, that's how we know where the start of the gene is, and therefore where to begin making your mRNA. Because remember, your DNA molecule is long, the gene is only a section of that. So how do they know where to start, and so on? And so in part that's with the codons here. And so your polypeptide, the first one would be methionine. So this is your first amino acid. So then, using the same thing, go and do your next codon here. So pause it and do that, and keep on doing that until you get your amino acid chain. So read the next codon and say what amino acid is, read the next codon, and so on. So pause it and do that. So when you paused it, you should have gotten the next one, ACU. So here's A, here's C, and here's U. ACU stands for the amino acid threonine, or THR. You don't have to memorize these, and you don't have to memorize the chart. And then the next codon is CAG. So we go, here's C, here's A, and G. All right, and so it's GLN. And then the last one, UAA, you should have gotten here. Here's your UAA. Notice here it stands for stop, and there's not an amino acid. So that codon doesn't stand for an amino acid. What that codon does is trigger your ribosome to know, hey, we're at the end of the mRNA, we're at the end of building our polypeptide, and we're done. And so this is your final polypeptide. And I just made it short. Obviously, these would be much longer, like 100 or hundreds long. But so that's a very short polypeptide, but that's how we know. All right, and so that's how it's built, and that's how translation relates with transcription and so on. Which, by the way, I wrote, instead of transcription, I wrote translation here. Keep getting ahead of myself here. Transcription, translation, and then your final polypeptide. So the genetic code, if we go down here, is said to be redundant. Why? More than one triplet codes for the same amino acid. So notice here, you have multiple ones that stand for arginine, multiple ones for lysine, threonine, and so on. So you have all these different ones that stand for the same amino acid. And so, but only one triplet codes for one amino acid. If you notice here, they call this the wobble effect. If you notice, like if you look at all the ones that stand for serine, what varies is the third base. Notice that the first two, UC, are U and C, and then the last base varies. Same thing with proline, it's CC, and then the last base varies. Same thing with threonine, AC, and the last base varies. They call that the wobble effect. That third base doesn't seem to play that much of a role in the amino acid that it codes for. All right, and so therefore that can change and doesn't change the amino acid in most cases. All right, not in all cases, but in most cases. So that's how we get multiple codons coding for the same amino acid. So then we write here, so then the reading frame is a really big deal. The reading frame is important because any error in the reading frame could code for gibberish. So if this is the 3 amino acids you're supposed to hook together, can you see that if you didn't read it AUG, then ACU, and you started here with U and UGA, that would code for a totally different amino acid, and then CUC, totally different amino acid, and you would get a different protein. So therefore, if you have your DNA mutated, so if this T mutates to an A and changes to an A, that's going to make this change to a U, and then the codon is going to be UUG instead of AUG, and you're going to get a different amino acid, which is just going to give you a different polypeptide, and therefore a different trait, and therefore that polypeptide might not be able to do its job in your cell. All right, and then you have side effects. So the reading frame and getting those 3 in the groups, the right groups of 3, is really, really important to hook together the right amino acids to make your right polypeptide in the end and your right protein. So let's look more specifically about transcription. So the gene determines the sequence of bases along the length of your mRNA molecule. So when you make your mRNA, going back over here, it's saying the gene in your DNA determines what the order of the bases in your mRNA molecule is going to be, which then is going to determine your polypeptide. Notice that when we used this chart, we used the codons in the mRNA. We're not using the DNA, because remember, the mRNA goes to the ribosome and is used to make your polypeptide chain. So then only one of the 2 regions of DNA serves as the template. So remember that DNA is double-stranded. So DNA, here's your double-stranded, here's your gene here. Notice that when we wrote the DNA, we're only using one side of the DNA. And we're looking at the 3 to the 5 prime direction so that your mRNA can be built in the 5 to 3 prime direction. All right, so we're only using one side. That one side is going to act as the template. And then DNA is red in the 3 prime to 5 prime direction. And that's the reason why, is so that mRNA can be synthesized in the 5 to 3 prime direction, just like DNA replication. All right, so let's look at then what we just talked about here, the translation. The mRNA triplets are called, what are they called? They are the 3, they are codons. Codons are written in what direction? 5 to 3 prime, that's how we write them and that's how we read them. They're read in the 5 to 3 prime direction along the mRNA, and the appropriate amino acid is incorporated into the protein according to the codon on the mRNA. So you read it from 5 to 3 in groups of 3 called codons. AUG stands for methionine, ACU stands for threonine, and so on. And as this is done, the protein begins to take shape. You get your polypeptide chain. Eventually, it'll curve on itself, and the R groups will interact, and the protein will get its ultimate shape that it will have at the end. So flip it over here. So we're going to look more specifically about how it is that we make that mRNA from DNA. So we're going to look at more specifically at transcription. Obviously we have to untwist and break apart the 2 strands of the DNA just like we do in DNA replication. But we're going to build an mRNA strand just from one side of the DNA. So let's look at how that happens. So mRNA and this enzyme RNA polymerase. mRNA is the messenger or vehicle that carries the genetic information from the DNA to the protein synthesizing machinery. What is that? Your ribosome. All right, now we have an enzyme that helps to make your mRNA molecule. So we have what's called RNA polymerase. Sounds very familiar to DNA polymerase. DNA polymerase 3 was used to make and add nucleotides to replicate DNA. RNA polymerase makes RNA. So the RNA polymerase is just like DNA polymerase, but it doesn't need a primer. So that's something different. All right, so how does this happen? So it's actually DNA polymerase 2. There's lots of different, there's just like DNA polymerase, there's lots of RNA polymerases. The RNA polymerase 2 is the one that encounters what's called a promoter region, promoter on the DNA, near a transcriptional unit and starts synthesizing and making your RNA. When the polymerase, RNA polymerase 2 encounters a terminator sequence, transcription stops. So on the gene, you have a promoter region that says, hey, there's a gene. This is where we're going to start transcription. We're making your mRNA. How do we know when to stop making the mRNA? We reach this terminator. All right. So when we look at this, look at here. Here's your RNA polymerase right here. That's your enzyme. Here's your double-stranded DNA. But notice here that this is the transcription unit in dark from right here in blue to right here. That's your transcription unit. This is what you want to make the mRNA from. All right, so this is the gene that's going to be used to make your mRNA. That's what we're going to transcribe. So that's your, all right, but look at here. The start point is right over here. All right, here's the start point. And so this is how RNA polymerase knows how to bind, is this promoter region. So it's a little bit ahead of that transcription unit, and it binds to that promoter region, recognizes that promoter region, and binds to it. So we're initiating transcription. Notice here that the RNA polymerase unwinds the DNA. So notice here, unwound DNA, here's your RNA polymerase, all right? RNA polymerase does this. Does this. So it unwinds the DNA and starts to make an mRNA transcript. So notice here your RNA transcript. Notice here it's building right here. And so notice it's using the bottom side of the DNA. Why is that important? Because it needs to be built in the 5' to 3' direction. And notice the bottom of the DNA, this is the 3', this is the 5', so it has to be anti-parallel, just like in DNA. So it's going to build and just use the bottom side as the template. And so then, notice here that RNA polymerase keeps on moving down the DNA molecule. And when it's done with that particular section, the DNA rewinds and winds back up and does the double helix. And notice that the mRNA, they call it the transcript. This is your mRNA, and it's kind of hanging off, and it's still being built here in the 5' to 3' direction. And so then it keeps on doing that until eventually it reaches that terminator sequence, and you have your completed RNA transcript. Now, realistically, this completed RNA transcript is only completed in prokaryotic cells, because in eukaryotic cells, so this is, and it's completed in prokaryotic cells here. Realistically, this RNA transcript is going to go and do RNA processing, which is that other step in between transcription and translation in eukaryotic cells. Okay. So if we turn over the sheet, we have a picture here. I meant to delete this. We don't really need this picture, so I'm not going to talk about it here. So we're just going to go right on to here. There's different types of RNA polymerase. So I alluded to the fact that what makes the mRNA is RNA polymerase 2. So prokaryotes only have one type of RNA polymerase that synthesizes mRNA, and other types of RNA as well. What do I mean by other types of RNA as well? We're going to learn that there's ribosomal RNA and there's transfer RNA, or tRNA. So rRNA and tRNA. And RNA polymerase makes both of those, but there's only one type in prokaryotes. That's different than in eukaryotes. Eukaryotes have 3 different types, and mRNA synthesis, as we said, uses DNA polymerase too. All right, so I just want to show that there's different types of RNA polymerase. So how in the world do we know what let's talk a little bit about that beginning promoter region. So promoters are found on the DNA molecule, remember. This is how RNA polymerase knows where to start making the mRNA molecule. So the promoter is found on the DNA molecule and initiates the transcription of the gene. So making the RNA, the mRNA molecule from the DNA. And this is the site where things that we call transcription factors and RNA polymerase, the enzyme that's going to make the RNA, and RNA polymerase attach. Transcription is finished when the RNA polymerase reaches do you remember what it is? What's it called when it reaches the end? It's the terminator. And so the stretch of DNA that's actually transcribed is called the transcription unit. So we looked at this on that previous picture. Usually extended a few dozen nucleotides upstream, so that's before, from the transcription starting point. That's what the promoter is. So this is all about promoters. Promoters include what we call a TATA box. TATA because the order of the bases are TATA, and this is in eukaryotes. So promoters are important for the binding of RNA polymerase, so we know where RNA polymerase knows where to bind and therefore to start transcribing the genes. And so let's look at this, initiating of transcription, which is where that promoter region is on the DNA. So these transcription factors, which we'll look at in just a minute in a picture. So these transcription factors bind the promoter region and therefore enable the enzyme RNA polymerase to do that as well, to bind. So the RNA polymerase 2, I'll just put RNA polymerase 2, binds with additional transcription factors, creating what's called a transcription initiation complex. And what happens when it binds there is the DNA unwinds and transcription begins. So if we look here at this picture, so what are we looking at? This part right here, this is your template DNA strand. So this is your DNA. And so right here, this is the gene from right here that this is the part we want to transcribe right here. So that's where we want to make the mRNA from. So how does RNA polymerase know where to bind? So when we wrote here upstream, a few dozen nucleotides upstream is where we find the promoter region. So here's the promoter region right before the part that we want to transcribe. And notice here the first part of the promoter region is the TATA box. So right here and right here is the TATA box. This is part of the promoter, and the TATA is referring to the order of bases. All right, and it's part of that promoter region, and this is what helps the transcription factors to bind. So here we have transcription factors. These guys here, transcription factors, I'll put in purple. All right, what are these guys? All right, let's write here that these guys are a collection of proteins that help RNA polymerase, let's put RNA poly, to bind and transcription to begin. All right, so what they're not showing you here, so the transcription factors bind, this is right here from right here. This would be the whoops, I changed colors on you. This right here would be the TATA box here. So this is the TATA box. And that's, so it's helping to know for the transcription factors to bind, okay? And so we can say here that that again, in addition, the transcription factors, they recognize the TATA box and binds to it, allowing, why is that important? Allowing RNA polymerase to bind. So if we look here from here to here, now you can see the transcription factors are still there. All right, so here's the transcription factors. You can barely see them on your copies there. But then once they bind to the transcription factors bind to the TATA box, now your large RNA polymerase enzyme can bind. So that's RNA polymerase 2. Notice here, then this was here's the start point right up here, and here's the start point for transcription. Here's the start point right here. RNA polymerase unwinds the DNA in just in a certain section, and you begin making the RNA transcript, which is right here. So this is your RNA transcript, and is made in the 5' to 3' direction. So here's the 5', it's going to be the 3', and this is why we're only using the bottom side here, because the bottom side of the DNA is 3', and it's got to go in the opposite direction. The new RNA is made in the opposite direction. And that's how in the world RNA polymerase knows where to actually start transcription. It has to do with those TATA boxes and the transcription factors, so it knows where to bind and therefore start transcription. All right, and that's called the initiation of transcription. So it has been started, it has been initiated. Okay, so then flip over. What the next picture does is enlarge this particular part here, RNA polymerase with the DNA and the mRNA that it's making. So when we look at this picture here, so here, going back from the last picture, here's your RNA polymerase. It unwinds and breaks apart the two sides of your DNA, so this side and this side. This side is the template strand. It's the 3' end of the DNA, and so the mRNA then is going to be made in the 5' to 3' direction. So this is showing you RNA polymerase, as it moves along the DNA molecule, it's going to unwind and untwist and break apart the two sides of the DNA and add the appropriate nucleotides. These nucleotides are found in the cytoplasm, so floating around, actually in the nucleus, which is where mRNA is made. So you have these nucleotides. What is a nucleotide? The phosphate, the sugar, and then the base, and then remember we have A's used for RNA, and C's and G's. So this part right here would be the phosphate sugar backbone, and it comes and binds with the complementary DNA. So you're making the mRNA in the 5' to 3' direction. So 3' end, 5' to 3' direction. So it's continuing adding, and the mRNA polymerase will move along the DNA, untwist it, and break it apart, and keep on adding nucleotides. After it's done, the mRNA is made. This growing strand kind of hangs off, and as the RNA moves, the RNA polymerase moves to the right, the DNA will rehydrogen bond back together and twist back up, and so you only have the part open that the RNA polymerase is working on at the time. So let's look at this difference in that, what we just talked about, the promoter region, how that's different in prokaryotic versus eukaryotic cells. So in prokaryotic cells, RNA polymerase recognizes and binds to the promoter on the DNA immediately and begins synthesizing mRNA. All right, so that happens immediately. In eukaryotes, a group of proteins called transcription factors are needed for the binding of the RNA polymerase and initiation of transcription. So what does that mean? In prokaryotic cells, there's no transcription factors. Once the transcription factors bind to the promoter, then RNA polymerase II binds and the transcription can proceed, just as we looked at. So the entire group of proteins in the eukaryotic cell that are involved in this are called the initiation complex. So they're called the initiation complex. So this whole thing with the promoter, the transcription, put this so you can see it, promoter, the transcription factors, RNA polymerase, all of those involved in starting transcription, it's called the transcription complex. All right, and it's a lot simpler in prokaryotic cells. All right, so transcription, so we just looked at how transcription starts. Now we're going to look at how it elongates. So as the RNA polymerase moves along the DNA, it uncoils it, we just talked about that, synthesizes the mRNA transcript, and peels away the DNA, allowing it to recoil after it's done. So it rebinds back together, twists back into the double helix. And just like before we talked about, there can be numerous RNA polymerases that can transcribe the DNA segment at the same time. Let's cross out this. I don't know why that word protein is here. Let's cross that out. So what that means is that it allows the cell to make large amounts of protein in a very short period of time. So if this is your DNA molecule, your RNA polymerase binds at the promoter and then gets to the transcription unit and starts making your mRNA from that. And as that moves in this direction, a new RNA polymerase can come and start making an mRNA. So you can have many mRNA or RNA polymerases making an mRNA at the same time. So then what do I mean by that? So that's originally, this is RNA polymerase 1. So now as it's moving along, this is now moving along and it has your longer mRNA strand. And so then a new RNA polymerase comes and binds and starts making an RNA strand. And it'll move, and then another one, and another one, and another one. So you can get lots of mRNAs made from one gene. And then each mRNA, remember, can have many ribosomes transcribing it, or translating it at the same time. All right. So how does transcription end? So this continues on and on and on. And so how does it end? Remember, you get to that stop codon, and so you get to the end, or the terminator region. So in prokaryotes, transcription proceeds through a DNA sequence that functions as a termination signal, causing the polymerase to detach from the DNA. This release of the transcript makes it immediately available for use as mRNA. All right, so it gets to that termination, it detaches, and now you have this mRNA molecule, and now it can be used and go to the ribosome and be used immediately. In eukaryotic cells, when the RNA polymerase reads a signal sequence, it cleaves off the RNA from the growing chain as the RNA polymerase continues to transcribe the DNA. So even after it's done, it continues on, and it continues to read and transcribe the DNA and eventually falls off. This RNA produced that falls off is still not ready for use, and this occurs in eukaryotes, as I said. So why is it not ready for use? It needs to be modified. Needs to go through, RNA processing. Okay, or modification. So before that was when we talked about it gets shorter and we cut out some things. So what is modification? So the eukaryotic RNA transcript now gets modified before it enters the cytoplasm. The 5' end of the transcript gets modified before leaving the nucleus, and what happens is you get a 5' what's called cap of nucleotides. And at the 3' end, the 3' end is also modified. They put a lot of adenine nucleotides. This is called a poly, meaning many, poly A tail. So they put on a cap and a tail. So on this particular picture here, here's your DNA, and it's the pre-mRNA, so there's the cap and the tail that's added. So if we look at this and how that relates here, here's your cap, so that's what I mean by the 5' cap, and then here's your poly A tail. And so what, and this is the protein coding section, all right, and then they find that there's some, here's your start and your stop, so this is the start and stop here. They found that there's some other bases in there as well, and we'll talk about those a little bit later. All right, so this is what's going to actually be transcribed to make your protein. So what's the function of the poly A tail and 5' cap? Important functions of them. They facilitate export of the mature mRNA from the nucleus. Because remember, in eukaryotic cells, that pre-mRNA, the mRNA has to leave the nucleus, so they help with that. And they protect mRNA from degradation by hydrolytic enzymes. These are enzymes that do, remember, hydrolysis, which means that they are going to break something down. All right, so they keep it from being broken down in the cytoplasm. So they assist in the attachment of the ribosome as well. So they think that they help for the mRNA to actually hook to the ribosome to begin making your amino acid chain. So attachment of the ribosome to the 5' end of the mRNA. Okay, so that's going to be read in the 5' to 3' direction. All right, so that's the first thing in RNA modification. So that's the first step in RNA modification. The second step in there is talking about the shortening part here. So the mRNA is further processed after the ends have been modified. We add the cap and the tail. And this is called, what we're going to talk about here is called RNA splicing. The initial transcript, about 8,000 BP stands for base pairs, is reduced to about 1,200 on an average. So they cut out a lot of the mRNA to get that when we go from the pre-mRNA to the mRNA. And what happens is we have these large non-encoding regions. That means that they don't code for making a protein. They don't stand for amino acids to build to make your protein. So it's the non-encoding regions of the DNA that get transcribed. Those are spliced out, and so meaning cut out of it. And so since they don't code for making an amino acid, we cut them out. So that's what we see here. So a couple of things with this is that when we look at the pre, and this is the final, your mRNA final, all right, we add the cap and the tail, so we've talked about that. So we've added the cap and the tail. The second is the splicing, so cutting out the non-coding parts. So these two bullets here, I'm going to have us fill these in. Introns are regions that are removed. These are the non-coding parts of your DNA. So they don't code for making a protein. And the exons are the expressed regions. Those are going to code for making a protein. So these are your coding regions of your DNA. So notice here, when we look at a gene, look at your mRNA, so remember that you built this mRNA from a DNA. So we wrote here DNA here. So you had your DNA, all right, from right here to right here, before you didn't have the cap and the tail. So this is the beginning and the end of the gene, and the DNA would continue on. The beginning and ending of the gene. And so on this gene, you had exons, introns, exon, intron, exon. And so what happens here is that only the exons code for the gene. So whatever the order of the base is here, so A, T, G, G, C, and then you have the introns, and the introns don't code for making anything. So at the end of this exon, we want to continue on with this exon. So let's say that this was A, T, G, G, A, so C, C, A, T, G, let's say this is C, C, T, A, G, or G, C, A. All right, so let's say that that's it. So this is our introns and exons. So what happens is that when you make your mRNA, you code and you copy all of that information onto an mRNA strand. So, and when we make the mRNA, we make and copy all the introns and the exons, so the coding and non-coding regions. So for your mRNA here, let me pick a different color here. If this is the this is the 3' end of the DNA, this single strand, and this is the 5'. So we're going to build the mRNA from the 5' to the 3'. That's what it says right here. So then the order of bases here would be UAC, right? All right, and so go ahead and do that for the rest of it. Pause it and do that. We have C, U, G, G, U, A, C. Then we'd have G, G, A, U, C, G, U. So these are the order of bases here, and so then this is your exons and introns, and so on. And so what happens is only the exons code for making your amino acids and putting them in the right order to make the right protein. So therefore what we do is we get rid of all the non-coding parts, which are the introns, so we cut those out. So therefore when we notice here, none of the introns are here, we cut this and this out. So now the order of bases are just the exons. So it's UAC. We cut out the intron here, so the next base is GGUAC. We cut out this intron, and we have CGU. So that would be the order with the introns cut out. And so it's really important to do that, because if we left the introns in, remember you read in groups of 3, and it would screw up what amino acid is hooked to what amino acid, and you wouldn't make the right protein and the right polypeptide chain. So cutting out those introns are important. So how do we do that? RNA splicing, that's called RNA splicing, cutting out the introns, occurs via SNRPs. SNRPs consist of RNA and protein and together, joined together to form what we call a spliceosome. And this interacts with the intron to clip it out and join the exons together. So here's your intron and the exon. So we want to get rid of the intron. We're going to cut the intron out and join the two exons together. So here are your proteins. This is and your SNRPs. And come together with other proteins to form, all right, these two things together form your spliceosome. So those two go together. So this whole thing is a spliceosome here. And basically what you need to know is that the spliceosome hydrogen bonds and at the start and end of the intron, so this is your intron here, it cuts it out, snips it out, and puts together your exons together. And so therefore this is this part right here, this now right here. It's cut out, and so that's cut out. It splices together the exons. So we did this in different colors. This is one exon in purple. That's this exon right here, and this exon in blue. This is now this right here, and we no longer have the red. Intron. So it cuts out the introns, and so just like we did up here, now you have the bases in the right order to bring together the right amino acids to make your appropriate polypeptide. So why is RNA splicing so significant? In many genes, different exons encode separate domains of the protein product. So what does that mean? So remember, okay, so here we have, this is your long DNA strand, but just this part right here is the gene. And genes can have exons and introns, so we don't want to code for the introns, that's non-coding, and just the exons. So what happens is that this part right here, when you make the mRNA from that, and therefore make eventually your polypeptide, that part of the gene codes for making this part of your polypeptide. And then the intron doesn't code for anything, so this exon, let's put a different color. This exon right here, remember they're going to be spliced together and used to make the same protein. That's going to be this part of the polypeptide. That codes for making that part of it. And then lastly, we'll use blue. The third codes for this last part. And so therefore you have one polypeptide, but it has different domains or sections that might have different jobs, if this is an enzyme of the active site or something like that. So, so from your DNA, you transcribe, you make the mRNA, we process it, translate it, and so different exons correspond to different parts of your polypeptide. And so they think, so RNA splicing, and when we do this splicing, this processing part and cutting out the introns, the way the RNA is spliced determines which proteins will be expressed. Going back to this picture here, all right, the C and the U is considered an intron. The GGAU is also considered an intron. So what happens is sometimes we can change what is used as an intron. So if we kept this as an exon and didn't cut it out, then we'd have UAC, and instead of GGU for our mRNA, you'd have the C and the U in there, and you'd bring in another amino acid, which might make a different protein. And so what happens is some organisms splice RNA differently and thus translate the genes into proteins differently, contributing to differences. So some organisms have different ways to splice the RNA, therefore they treat different parts of the DNA as introns and exons than other organisms, and so therefore make different proteins, which give you differences. Also, the alternative splicing is one possible reason humans can get by with relatively few genes. So what we found is we make way more proteins than we actually have genes. And if one gene makes one protein, that doesn't make sense, because if one gene makes a protein, we should have just as many genes as we make proteins. And so what they think is that sometimes, going back to this picture, sometimes in making a certain protein, we treat like these two spots as introns, but sometimes maybe we'll make them all exons, and we therefore would have more amino acids that we'd hook together, and therefore might have a different protein. So we can use the same segment of DNA differently depending upon what we treat as an intron and what we treat as an exon, and therefore with a small amount of DNA can make a wider variety of proteins. All right, and so that's where I want to leave off today. Tomorrow we'll pick up with translation and how we actually go about using the spliced RNA so we don't have introns, and how we actually build the amino acid chain. All right, so you can look forward to that. I'll see you tomorrow.